Bee here, and welcome back to biology. Do you know the phrase carpe diem? It's Latin for seize the day. You may not know how to translate Latin, but someone somewhere did, and you can use the results of their translation to understand something written in a different language. Your cells have to do a bit of translating sometimes too. Remember the central dogma that defines the flow of genetic information as going from DNA to RNA to protein? If there was a language for DNA and RNA, its alphabet would be made of nucleotide bases. Seemingly endless combinations of A's, T's, G's, C's, and U's make what we would consider to be words. Protein, on the other hand, speaks an entirely different language. It is built from an alphabet of 20 amino acids, such as valine, histidine, and lysine. If RNA is going to be used to make protein, we will have to find some way to translate the nucleic acid language of nucleotides into the protein language of amino acids. The process of translation, as it is aptly named, does exactly this job. Any guesses where translation takes place? Do you remember the jobs of all the organelles you learned a few units back? Which one was our protein factory? It was the ribosome, so that's where all the excitement will be taking place today. After today's lesson, you'll be able to explain the difference between transcription and translation, construct a model polypeptide chain from an mRNA molecule, explain the steps of translation, describe the structure and function of the ribosome. So, we know that our mRNA molecule needs to be translated, but let's first review how we got that mRNA molecule to begin with. In transcription, DNA was used as a template to make a single-stranded mRNA molecule that left the nucleus and headed out to the ribosome. Because remember, the original DNA is too important to let it leave the safety of the nucleus. So, a cheap copy, the mRNA, is made and sent in its place. We said earlier that the bases A, U, G, and C are like the alphabet of RNA. It turns out that RNA words are made of exactly 3 letters each, and each word is called a codon. So a strand of mRNA with the sequence UCUAGGAUC would be read as UCU, AGG, AUC. Each codon can then be translated into a specific amino acid using a chart like this one. Don't worry, you won't need to remember which codons translate to which amino acid. Just know how to use the chart. Let's see if we can find our sequence on the chart and determine which amino acids it would code for. For the first codon, find U in the very center of the chart. Then go outward on the chart to find C. And then on the very outside, we need another U. This tells us that UCU codes for serine. We follow the same steps to find AGG, and we land on arginine. Our last codon, AUC, gives us isoleucine. So our mRNA sequence coded for the amino acids serine, arginine, and isoleucine. Sometimes you may see the codons presented on a table that looks like this, instead of the circular chart we saw before. The idea is the same. Find the 3-letter combination and see what amino acid it translates to. Let's try another example with this table. Let's find the amino acids coded for this mRNA sequence: UGU, AAA, GUC, GGC. You can use the color-coded keys around the sides and top to help you, or you can just scan through the table until you find the right combination. UGU corresponds to cysteine, AAA gives us lysine, GUC lands on valine, and GGC ends our sequence with glycine. We came up with two very different amino acid chains, just from a set of 3 or 4 codons, with 20 different amino acids to choose from, and some proteins being made of chains of thousands of amino acids. The possible combinations seem nearly endless. This is why proteins can be so varied and complex, and carry out so many different functions for our bodies. There are a few special codons to be aware of. The mRNA sequence AUG is called the start codon, and it signals the process of translation to begin at that point. You can remember that AUG is the start codon if you know the word augment, which means to add to something. AUG tells translation to start adding amino acids. There are also a few codons which tell the translation process that it's time to stop. UAA, UAG, UGA. The first step of translation picks up right where transcription left off, with the mRNA molecule being made and leaving the nucleus. Once it has reached the ribosome, each codon is found by and bound to the appropriate anticodon on a tRNA molecule. tRNA, or transfer RNA, is the second type of RNA that we have seen so far. On the bottom of the molecule is a set of 3 nucleotides called the anticodon because it contains the complementary nucleotides that will automatically pair with a specific mRNA codon. For example, an mRNA codon of UUC would bind to a tRNA anticodon of AAG. On the top of the tRNA is a single amino acid, and it just so happens that the amino acid carried on top is the amino acid that our table told us would be translated from the mRNA codon. So, the tRNA carrying AAG, which binds to UUC, would carry the amino acid phenylalanine on top, because the table shows us that the mRNA codon UUC codes for phenylalanine. Remember that to start the process of matching up tRNA molecules, the codon AUG must arrive. After AUG, each codon will match to a tRNA until a stop codon is reached. Once a tRNA matches to a codon, it adds the amino acid it was carrying to the amino acid that was carried on the previous tRNA. This way, a continuous chain of amino acids is formed as the tRNAs drop off their cargo. The growing amino acid chain is called a polypeptide. Transfer RNA, or tRNA, gets its name from the fact that it literally transfers amino acids to the polypeptide chain. You can think of tRNAs like little trucks, delivering amino acids all day long to the correct addresses. After the stop codon is reached, the polypeptide is released, and the mRNA molecule dissociates. After all, it was only meant to be a temporary copy, and its job is done. The polypeptide chain isn't quite a protein yet. It will need to get folded into a very specific shape, and much of this work will happen in the endoplasmic reticulum. Remember the ER from your organelles lesson? It's now time for it to get to work. The shape of a protein has to be just right for the job it's going to do, and the exact sequence of amino acids it was made from will determine the shape it folds into. So, being made of the right amino acids is critically important. You've now seen the whole process of protein synthesis. In transcription, an mRNA molecule is made from the DNA template. This happens in the nucleus. Then, the mRNA is sent out of the nucleus to the ribosome for translation, where the tRNAs bring the correct amino acid to match the mRNA's codons. Those amino acids chain together to make a polypeptide, and will get folded into the correct shape to carry out the protein's job. All in a day's work for your cells. Since the ribosome was the star of today's show, let's look a little more closely at its structure. It is made primarily of rRNA, or ribosomal RNA, and consists of a large and small subunit. The large subunit sits on top of the small subunit, while the mRNA strand being translated is sandwiched in between them. The growing polypeptide chain leaves the ribosome through the large subunit as it is made. Within a cell, some ribosomes are free-floating, while others are attached to the ER. This is convenient because proteins go to the ER to get folded and processed after the polypeptide chain is constructed in the ribosome. All cells contain ribosomes. In fact, some of your cells contain as many as 10 million ribosomes. No living thing can exist without protein. Therefore, no living thing can exist without ribosomes. In fact, even prokaryotic cells, which we generally say do not contain organelles, do contain ribosomes. Because even bacteria wouldn't be able to survive without a way to make proteins. We compared ribosomes to a protein factory back in our unit on cells, and now you know what exactly goes on inside that factory. As we went through today's lesson, we explained the difference between transcription and translation, constructed a model polypeptide chain from an mRNA molecule, explained the steps of translation, described the structure and function of the ribosome. So far, we've learned about 2 reasons why our cells have to make copies of our DNA. 1, to replicate for our bodies to grow and to replace dying cells, and 2, to produce proteins. A third reason we have to copy DNA is to reproduce, so that we can pass our DNA on to our children. This process is a lot like mitosis, which we've already talked about, but there are some differences too. It's called meiosis, and we'll talk about it in detail next time. Until then, remember, biology isn't just science. It's the way of life. See you next time.